So I'll be speaking to you today about the Alzheimer's disease sequencing project. And although there are some interesting scientific findings, I'm actually going to be focusing on kind of more study design and quality control issues around whole genome sequence data. But first, to give you a little bit of background about the study, in 2012, there was a presidential initiative focused on Alzheimer's disease, and the ADSP, the Alzheimer's Disease Sequencing Project, came out of that as a component of the National Alzheimer's Disease Project Act. And initially, there were 6 groups involved, although that has enlarged over time. But there were 2 independent genetic consortium involved in Alzheimer's disease research, the ADGC and the CHARGE consortium, and both of those consortium were the ones that provided the patient samples and the phenotype data. And then also involved were the 3 NHGRI-funded large-scale genome sequencing and analysis centers. NIA provides funding and project organization and support. And at this point, there are additional funded investigators who also participate. So the goals are, of course, to identify genes that are related to Alzheimer's disease, and specifically not only looking for genes that increase risk, but also for protective variants as well. There are different phases of the project, and so the data delivery has come in different phases, and therefore the data QC processing and analysis of course, occur in those phases as well. And of course, multi-phase projects present some challenges. So I'll describe those phases and then talk about what some of those challenges are. So the initial discovery phase had 2 components, a family-based study and then a case control study. The family-based study was whole genome sequence data that was generated on just under 600 individuals. And these were from large extended families that had multiple individuals with Alzheimer's disease. There are a few genes that are known to cause Mendelian forms of Alzheimer's. These families were screened and were not carriers of those mutations. The case control sample consisted of roughly 5,000 cases and 5,000 controls. And there is also the ApoE gene. For the E4 allele is a well-established risk allele for Alzheimer's disease. And in selecting the cases and controls, they were actually selected against carrying that E4 allele. Trying to find cases in particular that may have had a genetic risk or genetic burden that was not due to the ApoE4 allele. So the next phase was the discovery extension. And so let me also, back up and mention that the family-based study was whole genome data. The case control sample in the discovery phase was whole exome. Once that phase was finished, it was determined that we should not pursue doing whole exome sequencing, and so everything after that is whole genome data. So in the discovery extension, there were an extension of the family component. There were additional individuals from the original families as well as new families that were sequenced. And then for the case control sample, it was now whole genome data that was generated instead of whole exome, and trying to expand on the diversity of the participants. The follow-up phase is currently under process, and that will focus on whole genome data, again in case control subjects. And the augmentation phase is really taking other existing whole genome data sets and incorporating them with the kind of official ADSP project. So large project, lots of investigators, et cetera. complicated organizational chart, but I think one of the foundations are the different work groups where all of the investigators can come together and share intellectual thought. And so the work that I'll focus on today is coming from the QC working group, really trying to focus on the quality control of the data that then is going out to the other working groups, but also the public, because these data are publicly accessible. So for the discovery phase, the QC was actually done within the QC working group, and then subsequent to the discovery phase, a data coordinating center was funded, and so now the variant calling and QC and the actual work on the sequence data is done by GCAD. With input from the QC working group so that the protocols and what's going to be done is developed with this wide net of investigators contributing. So kind of where we are currently in terms of data across these different phases is the only whole exome data that was generated as part of the ADSP is this case control data from the discovery, again, just over 10,000 individuals. The original family data that was whole genome, and then the family extension, case control extension, and then giving an example of this augmentation phase. This other project, the ADNI project, has whole genome data, and we're looking to incorporate it and jointly call it with the ADSP data set. So here, in terms of whole genome, there are roughly 4,800 samples. One thing, though, that you can notice is that there are multiple sequencing centers. So these samples were split among the 3 NHGRI centers. The ADNI data was done at Illumina, and the platform has shifted over time. This, again, is just a different representation of the data that's also showing the different ethnicities. So the majority of the earlier data was European ancestry with also some Hispanic data. And now in the discovery, extension, and subsequent phases, again, a goal is to increase the ethnic diversity. Just to point out, there was this one African-American family. They were actually initially mislabeled as Caucasian individuals, and in the QC process, the fact that they were not Caucasian became very apparent, and so we were then able to go and check and just identify that there was an error in the data handling. So always learning things as you look at your data. So what is the process? We've already gone through the whole calling and quality control in the discovery phase, and those data are publicly accessible. Either you can get the BAM files or you can get QC call data. And they were called by 2 different callers, performed at the Broad and at Baylor. And then moving forward with the combination of data, we're still going to be using 2 callers, and I'll talk about that in more detail. And that's going to be performed, again, by the GCAD, by the data processing group. So when I go into detail and talk about it, I'll talk a little bit about what we did in the discovery phase, which was kind of our first try at QC of these data. And then as we're moving forward, we have already done some calling in QC of this first set of family data, which was the discovery and the discovery extension, just the family data. And then we'll talk about what are our plans for this set of roughly 5,000 whole genomes, which is ongoing. So we have a paper that's describing this whole process. It's on bioRxiv, and it's actually in press at this point in Genomics. So if you're interested in details, that's a resource for you. So basically, kind of a flow chart here is that, again, we had the data called on 2 different callers, so GATK and ATLAS. Each of those pipelines had specific recommended QC steps, and so those were implemented. And then once we did those steps, we did a further kind of very basic QC of removing monomorphic variants, missing rate, extreme read depth, and excess heterozygosity. Typically, you might think of that at Hardy-Weinberg equilibrium. We used a slightly different approach because it was family data and implemented a score test that would still be appropriate, even given the relationships there. And what we then did at that point was try to put these two separately QC'd sets of data together in what we called a consensus data set. So here, what we have is a comparison actually of genotypes split out by SNV variants and indels, and how, when we did that comparison, they fell out. So for the single nucleotide variants, roughly 91% of the genotypes were concordant between the two callers, and there was actually a very low percent that were discordant between the two. But what you did find is that there were variants that were called only by one caller or the other, and those represented roughly 9% of the genotypes over all of the calls. There was a small fraction of a percent of genotypes that were missing in both, and then there was a very few, it was only like a few hundred variants where they were called by both callers, but actually had a different alternative allele specified. The situation, however, as you can see, was different for indels. Indels are a little trickier to call, and so the concordance rate was much lower, and the indels that were called uniquely by the different callers was substantially greater for the indels than for the SNV variants. So one of the things that we were interested in is how helpful is it to go through this process of two callers, and there's a number of different things that you can look at, but we had family data, and so we wanted to take advantage of that. We did some kind of straightforward looking at Mendelian consistency checks, but we were also able to do kind of a more sophisticated look at this, and this is work that was done by Ellen Wiseman at the University of Washington. So these families, from each family, a limited number of individuals were chosen for whole genome sequencing, but DNA was available on a much extended parts of the pedigrees, and they typically had GWAS information available. And so what was done was that samples of inheritance vectors based on the GWAS markers in this larger set were generated, and then those inheritance vectors were used to impute the whole genome sequence variants in those individuals that had the GWAS but not the whole genome sequence data. And then the Mendelian consistency was evaluated. And so for each whole genome sequence variance, Mendelian consistency rate was computed per pedigree and then averaged across all of the pedigrees. And that's something that is called, in this scenario, an imputation rate. And a high imputation rate matches up with high Mendelian consistency and is taken as evidence of high quality genotypes. So here are some plots that are showing the cumulative distribution of this imputation rate. And the solid line here that is showing the kind of best rate are those concordant variants. So those are ones that they were called by both callers, and we kept the genotypes that were concordant, and those showed the highest consistency when looking at this imputation. The next line is actually the ones that came out of GATK, and the last line here are the variants that came out of ATLAS, and then this broader dashed line is the combination of the uniquely called one. So it's just combining the ATLAS and the GATK line. So I think one message is that overall the imputation rate was, or the Mendelian consistency rate was good. It was clearly best in the variants that were concordantly called by both callers. The imputation rate is also going to be dependent on allele frequency. And so the number of heterozygotes is an indication of that. And so when you have a higher allele frequency, you're also getting a better imputation. This data is publicly available, so others have used it. And here's a QC paper published by somebody outside of the ADSP investigators. They looked at the whole exome data and also, totally independently concluded that use of multiple pipelines is worthwhile. So we're trying to figure out, well, what do we do moving forward to take advantage of what we learned in the discovery phase and continue generating high quality data? And the challenges are we now have 2 different platforms and 4 different sequencing centers. And when we go to our next wave of data, we're going to be adding a new platform, NovaSeq, and a new sequencing center. Our plan is to jointly call the data sets across the centers and platforms to use the 2 different callers and create a QC consensus data set. What was found in that independent paper, and they looked at both aligners and different ways of implementing callers, either a single sample caller that's using data from a single sample at a time versus multi-sample, which is using information across samples to inform the calling. And they found that you get different variants called with those 2 different approaches. So I think GATK is a caller that people are familiar at. We're at the Broad, people way more expert than me in the room. So just to mention that XATLAS is a new release of the ATLAS caller. It does SNVs and INDELs, and it's very fast. So it can call 30x WGS on 1 CPU in less than an hour. And that's an important consideration because it does take a lot of computational time, and that does equal money, to generate multiple call sets. But this is looks like a feasible option because of the efficiency. So one of the other things that we've done is a replicate analysis. So for all of the ADSP data, 3 individuals have been sequenced on every platform and at every center. They happen to be a parent and 2 offspring. And so what we've done is done a principal component analysis to look at what is the behavior of these replicates through the QC process. So what you would expect when you're doing a principal component analysis is for those replicate individuals to fall right on top of each other. And when we look at the first principal component versus the second in totally raw calls coming out of GATK, you're seeing that happening. However, when you look at the 3rd and 4th principal component, you see total separation by platform. Okay? As you do QC, and so for GATK, you take the variants that pass. You start to see, with the exception of one of the sequencing centers, that the replicates are starting to collapse and come back together, which is what you'd like to see. Perhaps not surprisingly, we are not seeing this occur in the same way with the indels, which we know to be less reliable. And so here, with no QC on the indels, principal component 3 and 4, you're seeing this complete separation where the solid are HiSeq X and the open are HiSeq 2000. Even when we look at those variants that pass, we're still seeing that separation. And if we additionally take only those variants that have at least a 95% call rate, we're still seeing that separation. Some good news is that these principal components 3 and 4 are only explaining about 3 to 4% of the variation. So it's not like it's explaining a tremendous amount of variation, but again, I think that this is something you need to be aware of, particularly if, say, your cases and controls are not equally distributed across the platforms. This is the kind of thing that's going to lead to false positives when you're doing association results. So the next step in the data is to call these 4,800 sequences together. Again, that's ongoing. And so I think there are things to think about. What are the impact of calling these distinct data sets together? I think it's a good idea to include replicates in data whenever possible. There's tons of large-scale sequencing going on, coordination across those studies. If they could be using the same replicates, that would be really useful for when you're trying to combine data across ADSP and TopMed and CCDG, et cetera. Trios can be useful, or throw a few family members in there so you can look at things like consistency, Mendelian consistency. And of course, always look carefully at your data and your analysis results. We have identified some novel genes. In our next phases, we'll have even more power with our larger samples. The effort that goes into collecting the patient information, the phenotype, and doing the sequencing is tremendous. So apply thoughtful QC and calling so that you make the most out of the investment that's already there. Our data are publicly accessible. So if you'd like to download them, you have to go through the data use agreement process, et cetera. And of course, I am representing the work of a large number of individuals who've all contributed to the ADSP project. So thank you.
Thank you. Questions for Anita? A lot of things to remember to put into your statistical efforts. I'm not a statistician. Go ahead.
Have you looked at deep variant at all for your variant columns, other alternative algorithms?
No, we have not.
I have a question. I'm originally from Colombia, where 20% of the population is black. So our genotyping and sequencing data is closely related to the African ancestry. When you choose to work with ethnicity as opposed to race, like you showed here, how do you make sure that you can correctly stratify that data in order to avoid providing misleading recommendations? Does that make sense? I'm just saying, like, not all Hispanic data can be just associated with a particular race because it's multiracial, multicultural.
Right.
And black people from that particular region are most closely related to the African ancestry.
Right. So when you're trying to appropriately handle ethnic diversity, you have different sources of information. So one is self-reported race, and so we have that, and we have it broken out the way NIH does by both race and ethnicity. But the genetic information is what's really informative. And so there's a variety of approaches. So I showed here principal component analysis, trying to look at technical artifact, but of course principal component analysis can be used to capture population substructure. And then other investigators, some listed here and some of their colleagues, are also doing more sophisticated admixture work where you can look along the genome and identify which portion for an individual is coming, say, from European ancestry versus other sources. So I think it's important to look at that carefully, and there are many methods to do that and that are being applied. Does that answer your question? Yeah.
One here. Go ahead.
Great presentation.
Thank you.
It's very sad to see these discourse between these two software very big. I was wondering if you have these 6 samples with different platforms. You did some standard sequencing confirming the difference, calling the scores to see which one one is better or which one you have to trust more? Or, I don't know if you can talk a little bit more.
So there was some Sanger sequencing done to validate some of the top hits that we were initially seeing. And as it turned out, there were some issues in our first set of calling because they weren't, the files, so the calling was done jointly, but there was some processing that was done at different steps. And so different alignment files had been used. And so it was actually generating artifact. So for some of our top hits, sequencing was done, and it actually helped to determine that there was this issue. There has not been larger scale sequencing to kind of follow up those differences. I know I have a grant in, and if it gets funded, there would be some additional sequencing done in terms of validation work, and so I'm sure other people are working on that as well. Yeah.
Really interesting sequencing 6 samples from platforms and scores between the two. So, you know, only to measure much more.
Benchmarking.
Right, right. Right. But even without the sequencing, I think, again, when you have the family data, the Mendelian consistency or inconsistency is also really helpful in terms of determining what are valid calls and what are not.